I close my eyes and let my heart go back in time, you're still here, like you were yesterday. I can make your heart stand still. I'm the only one who ever will.
Hi everyone, how have you been? So today we are jumping into a new chapter which deals with the DNA interaction. Actually, the genes interaction, should I say, to finally produce the protein which make possible for every different forms of lives. So this chapter inevitably contains some of the technical descriptions. So whenever possible, I'll try to explain uh, as much as possible so that in a simpler form. So even those non-science major can hopefully understand what's going on in the modern DNA technology. Obviously, I cannot cover everything, but at least uh, some of the important and also up and coming the new technology which will change the way we live, the way of life in the future. We also try to at least briefly cover at least so that you can understand basically what are they type of things. So DNA is actually nothing but an information. It is not. It's a conceptual things, and that conceptual things will be converted into the realistic, tangible thing. Finally, when those information contained in DNA is converted into proteins. That's how we can see the relationship between DNA and protein. Obviously, there is a DNA and there is a protein. And then in these days, DNA, the information, this is a simply the information. And protein is the workhorse. And there is a middleman which delivers the DNA information. It's like a blueprint. DNA, but not every DNA is, but only some information contained in the DNA. So those information that is used to become the protein we call as a gene. Of course, we are going to have a more accurate definition of the genes in this chapter when the time comes. And then RNA get this in the form of copying that information and then delivers uh, that information in the process of the protein synthesis. Okay. So this from DNA to RNA making process we call as a transcriptions. Whereas from RNA to protein, this is actually a conversion process. Up until this is simply conceptual information stage. Then only actual tangible things is made only after protein is built based upon that information. Okay. And because of that, this process from RNA to protein is called translation. So So here we are going to have a brief survey of to begin with the organization status and the WAS gene and how the RNA is made from genes, what can control that activity, because that is extremely important activity. So definitely we need some of the fine control mechanism. Okay. And finally, how that information is converted into protein, and what happens if that conversion process has gone something wrong. That is like where we can understand the basic nature of mutation, is how this chapter is organized. So first off, here we are mainly going to focus on the eukaryotic genome. Not that the prokaryotic genome is less important or there is not much complicated issues involved, so we don't really need to take a look at. No, not at all. But simply the time conflicts, the time limits. Since there's no way we can cover everything, so we just selectively only concentrate rather on those issues and stories about the eukaryotic genome. That's how I chose to have this lecture organized. By the way, today's lecture I opened up the window of my room, so it may actually have a little bit of the nuisance, but distractions, should I say. But probably it's better to have a fresh air while it's just for the sake of my own selfishness. So bear with me if you are kind of a little bit bothered by potentially some noise coming out of outside. Okay. So eukaryotic genome is organized in the chromosome. We already know that. We have briefly at least looked at it in the previous chapter. So for example, this human genome, speaking of our own genome, if we line up all the DNA, which is made up of double stranded, as a result of the complementary base pairing, it consists of about 3 billion such nucleotide pairs. So those order of 4 different kind of nucleotide pairs are basically what you can say is in random order. So because of that, the certain segment of those DNA that we define as genes, the DNA sequence. DNA sequence, what I mean by DNA sequence is the specific order of such nucleotides are random, and the uniqueness of each different genes can come from such organization scheme. So it's about 3 billion nucleotide pairs, right? And I have already mentioned that earlier. That's long, such a 3 billion base pair long DNAs has been condensed to such a compact structure. It's like a remarkable job of how those long such DNA strands can be compacted into such a small, tight structure that can fit into a very small area of nucleus of the cell. Anyway, we all know that such 3 billion base pair long DNA is not does not exist in just a lengthy one linear strand of DNA, but the DNA is divided into several segments. That organization unit we recognize as a chromosome. So we do have 23 different such chromosome in our cell. Okay, but usually we describe the status of those chromosome organization in this way, like 2n equals 46. What does this mean is this 2n equals 46, but actually the number of unique chromosomes in human cell is only 23, means this is a diploid organism. Diploid means we have already looked at in several occasions that I try to distinguish the difference between diploid and haploid. Diploid means having two sets of chromosomes. Two sets of chromosomes. So we have 23 kinds of chromosomes, like such that we can number each different chromosome, like chromosome number 1, 2, 3, 4, 5, 6. You can go all the way up to 23. But the 23rd chromosome, depending on who you are, it could be either an X chromosome or Y chromosome. Okay, you guys don't have any Y chromosome, but I do. Luckily or unfortunately, I do have a Y chromosome. But that is a 23rd chromosome in such organization scheme. However, in every different normal body cell of human, normal body cell means normal. I shouldn't say normal. What do I mean by normal? That is actually the one of the most awkward vocabulary. Normal. So in body cell, regular or standard body cells, in the professional terms referring to such body cell, it's a somatic cell. We have two sets of such 23 different chromosomes, so altogether total number of chromosomes is 46. However, they are in the duplicated status. Why? Because this somatic cells, the origin of somatic cells is a zygote, which is fertilized cell or fertilized egg, either way. Let's just put it fertilized egg. And then cell division afterwards. Many rounds of cell division saw the origin of this somatic cell. On the other hand, that fertilization is actually coming from two different types of the haploid cell, the sex cells. Gamete, which is unlike the somatic cells, sex cells or functions are only in the sexual reproduction. So only those who are involved in the sexual reproduction have the gamete. So the example of gametes, egg cells, and sperm. They are, unlike the somatic cell, these gametes are haploid, which means having only one set of chromosome. So because of this one set containing an egg, another such set containing a sperm, combined together during the fertilization, and then you restore in terms of diploid status you restored in the zygote. So that's the relationship between haploidy and the diploidy. So getting back to this description of 2n equals 46, when usually certain the genome status was mentioned in this way of 2n equals 46, then you know, okay, this is referring a diploid cell. On the other hand, if this was meant to mention an haploid status of a cell, then instead usually you should describe this as n equals 23. Okay. So in other words, here n by definition can be said of number of unique chromosomes of a species or an individual that belongs to the particular species. So n can be considered to be equal to haploid chromosome number. And obviously that n number, specific n number, is different in different species of the organisms. So for example, these lovely dogs, the species, dogs as a species, has 2n equals 78. So diploid number of chromosomes of dogs, whose scientific name is Canis lupus, right? But this Canis lupus cannot distinguish it from its ancestral form, who are the wild wolf. The actually gray wolf is considered to be an ancestral form of all the modern day different breeds of dogs. So that to make a little bit of a distinction, so in the case of dogs, this is called a subspecies name, familiar. Actually there is a typo, so instead of familiar, there is one I is missing. The Canis, so sorry about that. Canis lupus familiaris is its full scientific name of dogs. So here the point is, its genome size is very similar to that of a human. That means the entire DNA consists of about 3 billion nucleotide pairs, just like humans. However, they are, unlike the case of a human where those 2n equals 46, that means there are only 23 different unique chromosomes. But in this case, the entire genome, the diploid number is 78, which indicates that the average size of chromosome in dogs would be larger or smaller, okay, with the same size of genome, similar size of genome, being divided into more number of chromosomes. Okay, that's that kind of information we can get from this way of describing their relative genome size and an organization. In the case of this fruit fly, its scientific name is Drosophila melanogaster. Although these fruit flies are such a disturbing, unpleasant nuisance to us, but it is actually a very valuable, scientifically speaking, organism from which throughout the research of this fruit fly we have obtained huge amount of valuable information. Why this particular fruit fly has become it's called those kind of organism utilized in studying for the sake of learning more better about our own genome. They are called model organisms. A lot of model organisms exist, but one of very common characteristics for all those the model organisms are their genome size. If their genome size is very small, then it's much easier and convenient for us to have many diverse studies. So here, what we can immediately notice is their n equals. Here only the n has been described as a haploid number, but that doesn't mean the fruit fly is a haploid organism. Actually, fruit fly is also diploid organism. For the sake of convenience, to make a easier comparison, that's how I just simply decided to use this haploid number. So its haploid number of chromosomes is have only 4, which is very simple, right? But actually the number of genes present in this very small number of chromosomes is not really that proportionally smaller. It contains really decent amount of genes, which means most of those genes are something that relevant to they have a counterpart to the human genes. So that's the benefit of researching such a small size of the model organisms. In the case of the very renowned plant version of the model organism, Arabidopsis, which is a scientific name. I forgot its common name. Crephails? I don't know what's this kind of weird common names for the Arabidopsis. This Arabidopsis is just such a famous. Then usually people go by its scientific name, Arabidopsis, but it is such the wild small weed plant. But as I mentioned earlier in the previous chapter, out of this weed plant, many important crops, including the what was that? The kales and what else? Like cabbages and several of those very important the vegetables were actually produced through these artificial selections, as we have seen earlier. Anyway, this is another and one of the major model organism in plants. Haploid chromosome number is only 5. Corn, on the other hand, has 10 such haploid chromosome number. Tigers, 19 chromosomes. Wow. Okay. Lion, 19 chromosomes. Actually, both tiger and lion have 19 chromosome numbers. Signifies that something that may between the hybrid between tiger and lion may not, after all, be such an impossible thing. And then we learn that actually, they can produce a viable hybrid between tigers and lions. So in other words, there isn't actually the biological barrier or reproductive barrier between these two different species. It's only something like a geographical barrier prevented for them to have just a frequent gene flow to occur. That's why they have remained in a separate species. Otherwise, probably they would have been actually produced a viable hybrid, and so there wouldn't have been any clear distinction between these tigers and lions because their chromosome numbers are compatible. Hyena, on the other hand, has 20 chromosomes. Okay, and horse 32, and donkey 31. So this is actually the problem, as I've actually went over when we had this issue of hybrid compatibility in earlier chapters. That's why even though between donkey and horse can be forced to mate, but their hybrid, which is mule, has a problem of producing a viable gametes because of this unequal number of the haploid number. That's why they are sterile. Chimpanzee is chromosome number is 24. Like if we compare this as with that of humans N chromosome, the genome organization, N equals 23. So we just immediately know that it is physically, technically impossible between chimpanzee and human to have any viable offspring. Okay, that's it. And another thing about this chromosome number and the genome size is just because you have such a the high number of n value doesn't necessarily equals to their absolute genome size. Case in point is this. Another butterfly. I don't know why I love to mention butterfly. Maybe it's a symbol of a freedom. I don't know. The name for this particular butterfly is an Atlas blue, which is a resident of the northern North America. Its N number is like outrageous. It's about 224 to 226. It's been determined clearly exactly what is that. But anyway, it's around like something like 220 something number of different chromosomes. Can you believe this? Has such a huge number of chromosomes, but that doesn't mean that the absolute genome size of this butterfly is huge and like as huge as that of a human. Yeah, that is something that we should remember. And another thing is, just because you have such a huge genome size doesn't mean that you are a little bit like something an advanced organism in terms of the evolution, the lineage. So certain protists are generally considered to be like a little bit of a primitive in terms of relatively speaking primitive in the eukaryotes evolution lineage, but some protists have huge, several times as large as that of a human genome. So that doesn't apply. That just because you have a huge genome size doesn't mean that you are something like having larger genome size is equal to the larger number of genes. Absolutely no, that is not the case. So as I went over briefly, this is the relationship among the DNA and RNA protein. Okay, this type of relationship from the information here, the flow of information. Later, I'm going to mention once again, but this type of relationship is usually called the central dogma. which describes the flow of information, the direction and the flow of the information from gene to the protein. Here, another hidden importance of this, the flow chart, so to speak, is the information can only be transferred from DNA to RNA, this direction, which means not that direction. You cannot go reverse. Okay? And also same thing is from RNA to protein here, but not that direction. So this is always one-way direction from gene to DNA. DNA is the one contain the gene, although not every area of the DNA act as genes. So that's why we are going to define what exactly genes are shortly after. Okay. But this, whatever this central dogma, is actually we learn that certain organisms are simply capable of violating this dogma. Is something that a type of rules or principle that you cannot violate is usually called a dogma. So in terms of that, this is not a dogma anymore, because we now know that certain viruses actually, at least, can simply move backwards. They can make their DNA from starting from the RNA. And also it is not entirely, we have recently learned that it is not entirely impossible to generate RNA starting from protein. So what's central dogma? It's just like it reflects only the earlier days of this level of the studies in this area. So anyway, the DNA is we all know that the long chain of a polymer made of 4 different kinds of nucleotides, which is a nucleotide but usually having 3 phosphate contains. We have already dealt with in previous chapters. So when we try to build this chain of a polymer, we usually what we have to take the form of nucleotide is not the one containing single phosphate, but we should have 3 different phosphate. And during this incorporation, what happens is you split this. So this is actually the hydrolysis or the catabolic reaction, and then only this part will be incorporated into in building this so-called phosphodiester bond to generate chain of nucleic acid. Okay. So the reason why we have to use this 3 triphosphate like triphosphate version of the nucleotide is by doing so, you are by breaking this phosphate bond, you can generate the energy. This energy can fund to build the long chain of this, the synthetic reaction of a nucleotide synthesis, so that there is no reason for you to provide any additional energy to this synthetic the anabolic reaction, which this issues appeared on your last midterm exam, right? So it goes like that. Okay. Here one information we can learn out of this way of nucleic acid synthesis is that if you keep doing this, what you can immediately notice is, so let's say this is a nucleotide number 1 and 2 and 3, and the direction of addition is always from this phosphate group after shedding the 2-phosphate group off. This unit will try to enter, okay, so this phosphate and the connection between this phosphate and there is a base will also be made. Okay, so it's always the addition of new nucleotide into the existing nucleic acid polymer is always from this. This is phosphate group of the incoming nucleotide to the existing. This is in terms of a carbon number of the ribose sugar. This is number 1 ribose sugar, and this is number 2, and this is number 3, and this is number 4, and this is number 5. Although this carbon number of the ribose is always, you have a prime, number 2, 2 prime, number 3 prime, 4 prime, and 5 prime, because you do also have a carbons right there. So in order to make this a separate distinction, in the case of a ribose sugar carbon, you put prime in instead. So in other words, this is always from 5 prime phosphate to the 3 prime hydroxyl group. Here the 5 prime means the phosphate group attached to the 5 carbon of ribose, and 3 prime hydroxyl group means the hydroxyl group attached to the number 3 prime carbon of the ribose. Okay, let me clear up this too, so that you can have a clear understanding of this. So the bottom line of this information is the number 1 nucleotide, you will always have this phosphate group, but this phosphate group is what? What is this phosphate group? This is carbon number 5 prime. And the last nucleotide, you still have this number 3 prime carbon. There is OH, the hydroxyl groups available, to which another new nucleotide can be added, right? And on the other hand, the first nucleotide, this is nucleotide number 1, okay, you do have phosphate group which is attached to the 5 prime carbon of the ribose. Is still available, it's a free, this phosphate group, and also this hydroxyl group is free on the other end of this nucleic acid, if you follow what I say up until now. So in this way, so this is 5 prime phosphate group, and this is 3 prime hydroxyl group. You can designate in between. If you examine in between all those nucleotides, whether this so-called 5 prime phosphate or 3 prime hydroxyl, all of them are, they are already used up, so they are occupied. The only 2 free are either phosphate or hydroxyls can be seen only at one end, which is now we can call as a 5 prime phosphate end, and the other one is the other side, the other end, is a 3 prime hydroxyl group. They are the only 2 free those functional groups. So in this way, we usually designate the directionality of any single nucleic acid. It applies to both DNA and RNA as well. Same thing goes for both of these nucleic acid. So we can make the direction when speaking of indicating the direction of any nucleic acid. We usually call this end as a 5' and simply the other one end as a 3'. And that is simply very convenient way of describing actual relative direction of a nucleic acid. And here what you can say is the synthesis of nucleic acid, whether it's a DNA or RNA, the synthesis always goes from 5' to 3' direction. And hopefully you understand the reason why, because the synthesis is dependent upon the formation of this so-called phosphodiester bond, which should occur between the existing nucleotide, the last nucleotide's 3' hydroxyl group, and incoming new nucleotides, the 5' phosphate group. So if you keep doing this, the direction of the new nucleotide addition will always move from 5 to 3' end. So that's the convenient way of describing the direction of nucleic acid. What's more important is the actual organization scheme of DNA double helical structure that can be said of antiparallel double helix. Here you can clearly see what this antiparallelity means. That is, here in this DNA double helix, how do you know whether it's a DNA? Because you see this T present, so this must be a DNA double helix. RNA can also make the double helix, although not as often as the DNA, and usually in nature RNA doesn't like to have the two different strands, the complementary base pairing based upon double strands. They rather actually prefer to have their internal complementary base pair, and that's the structural basis for certain RNA can act as the enzyme known as ribozyme. So anyway, here what you can see is on the left side of the DNA strand, the direction of this strand, it goes from here, the top is 5' end, right here, and the bottom is a 3' end. So you can describe this direction as in this way. Usually by convention, you put the arrowhead at the 3' end of any nucleic acid, whether it's a DNA or RNA. So here is a 5' end and this is a 3' end. On the other hand, if you look at this, the other partner strand of this DNA, it goes from bottom to the top. So here is a 5' end and the top is a 3' end. That's what I mean by the antiparallel nature of this complementary base pairing. So in the case of DNAs are held together in the antiparallel way. In the case of RNA, yes, those RNA double strand, if you ever discover them to exist, yes, that's possible, and then it also exists in antiparallel way. And what about the DNA and RNA hybrid? It is also possible. One DNA strand can pair with another RNA strand. In that case too, DNA and this RNA hybrid will be organized in the antiparallel way. It isn't like a requirement for their structural reason. When actually when Watson and Crick first identified to figure out the structure of DNA double helix, they had a last challenge obstacle to solve. No matter how hard they tried, they could actually make these two DNA strands perfectly fit. So it's their attempt, like in danger of a failure. But then they tried. How about this? So having one strand that like stand upside down, and then everything actually became fit perfectly into, because of their relative dimension of its nucleotide, the nitrogen's base over here. In that way only can make the respective hydrogen bond between two complementary nitrogen's bases. That's the reason why they have to have this antiparallel way of interaction. Although there is an exception, so you would in nature occasionally you could find some of the interaction between two DNA or two RNA or between hybrid in not in the antiparallel way, but that type of exceptions we better not mention here. We are only here talking about the standard way of the way they are in their existence. It's just like I said, nucleic acid are in the antiparallel way of their complementary base pairing, but then later you discovered some very few exceptions that are not in such an organization pattern, and then hey, you lied. No, it's not that I lied, but yeah, I just simply ignored a little bit of such very minor exceptions. So for those who are much more interested in such a higher level of in-depth studies, maybe as a major, maybe you can find out yourself. Okay, some other occasions, since they're not such like this standard way of antiparallel way. And this is a general organization structure of model figured out by Watson and Crick. However, actually, this double helical structure, the clue about DNA existing in this mysterious helical structure originally came from the work of this young lady at the time, young lady scientist. Her name is Rosalind Franklin. As you can notice over here, she had such a remarkable, brilliant, but unfortunately very short life. Yeah. And this is her work data, which, using the X-ray crystallography. Basically what it is you make the DNA structure in forms of a crystallize the DNA. Sometimes it's not really that difficult technically to crystallize certain material. Some other times it's much more challenging. But once you crystallize this any material, and it's very easy, I should say it's easy, it is possible to reveal its kind of basic structural organization. That's how. So after crystallizing this DNA, she subjected a DNA crystal into the X-ray beam, and this is a kind of a pattern of a diffraction. Basically what it is X-ray beam hit certain part of DNA structure, and then has a diffraction is something a little bit different from reflection, but so you have this kind of different angles, or due to the structural form, different angles at the back of DNA molecule. You place the X-ray film just like the same X-ray film you use when you take this X-ray examine for your health exam. Then you do have certain pattern, and by reading and interpreting the pattern, you can figure out the basic organizational structure of the material. In this case, oh, this means this regular pattern of this diffraction pattern indicates DNAs are in some kind of a helical form. And more detailed dimension of the structure, even I forgot. I used to know, but yeah, these days I easily forget something. But anyway, basically that's what it is. So she's the one, the pioneer that this, like at the time, the revolution it is around 1950 something, 4 or 5. That was then in Britain. So then she thought the DNA might be in the helical form. Possible option would be double helix or triple helix. This pattern actually could be true for both, either a double or a triple helix. So she tried to figure this thing out to resolve this by further actual experiments. Okay, so she's been working so hard, day and nights. In other words, actually, if you had to work in this type of experiment, and it is not, you cannot avoid having the too much of X-ray exposure, which is which have actually acted on really hazardous, put some hazard on her health, and that is the reason why she had such a short life. She died of cancer eventually, several years after. But anyway, to the Wasserman Creek. Fortunately, was able to have an access to her data without her knowledge, because Morris Wilkins was her boss in the laboratory, and Wasserman Creek were working together in a nearby in the research circle in Britain at the time. So Morris Wilkins, yeah, should I say like stealing the word is too strong, but basically what he did was a stealing. He stole her, the Rosalind Franklin's this X-ray crystallographic data from her desk, and then delivered that data to Wasserman Creek. And by looking at the data, they knew, okay, this is some kind of a helical form. And then instead of taking the standard step of to figure whether distinguish whether it's a double helix or a triple helix, Watson and Crick decided to jump onto already pre-established conclusion. Okay, let's assume this is double helix because we believe this is double helix. Sometimes actually in that works, although it is not, a lot of times it is not a recommended way of conducting the science, but like the Rosalind Franklin's way is actually a long way around, whereas this one, if they were lucky, which was the case, then they could have reached the conclusion, correct conclusion, get the result much quicker. So they decided to build the model based upon the assumption of that this DNA was in the form of a double helix. Okay. And they succeeded. Voila. So that's why they were able to get rewarded by the Nobel Prize several years after, together with Maurice Wilkins. What did he do? Yeah, just simply the contribution of the stealing the Rosalind Franklin's data and presented it to them. So three is the co-recipients of the Nobel Prize. What about the real guy, real woman, real lady, I should say, a real lady who should be recognized as the most contributor of this information, the Rosalind Franklin. Unfortunately, at the time of this award, she does not. She's not with us on Earth, so the Nobel Prize cannot be awarded to any those post-mortem any deceased individual. So that's the only reason actually she was not able to receive a Nobel Prize together with these I will just skip. I will skip the any adjective. These gentlemen. Okay. Actually, the hidden significance of this DNA structure, just because Watson and Crick were able to present correct structural model of DNA double helix, it doesn't warrant any such a great honor like Nobel Prize. By figuring out the correct DNA structure, actually we could learn a lot more significant information, including how DNA can replicate, and also the DNA is the actually genetic material. At the time, the majority of scientists believed that the protein was the genetic material, not the DNA, because obviously proteins are made up of 20 different amino acids, so it could generate more variants, which is significantly critical for acting as genes. But in case of the DNA, only 4 nucleotides, so a lot of people thought, hey, what can only 4 different nucleotides can make any kind of variation, if ever? And another fact actually clouded this the judgment was the fact that they discovered that in chromosome you have 50% of the protein and 50% of DNA. Okay, so which ones? Then a lot of these scientists believe that, okay, this DNA is such a very repetitive DNA nucleotide. It's just judging from the nature of the nature of DNA nucleotide, it must be a very repetitive, boring structure. So probably this DNA's role in the chromosome is a structural function to protect the valuable information contained in each different amino acid all together, acting as a gene. So, yeah. So it sounds funny here at present by how much silly can that be? They be, but just like all other science, we are still in the early infancy. Even though we may think that we know everything almost in these days, thanks to all those modern brilliant technology, no, we have, just like them in the 1950s, we have such a long way to go in almost every bit of this area of the life information. Okay, so this DNA, we have only 4 nucleotides, like I said here. But that doesn't mean that you cannot really generate any meaningful, significant variation. This random arrangement of the 4 nucleotide, how many? So, for example, like if any DNA fragment is made up of only 100 nucleotide, then using 4 different such nucleotide, the possible different outcome of the combination, in terms of combination, there is a 4 to the 100th possible different ways to assemble DNA fragment, because, right? So position number 1, you have a 4 different way. In position number 2, you have a 4 different way. Position number 3, you have 4. Just like that. And then it's a huge variation. So if you are a little concerned about like the number of variation that can be generated out of this nucleotide, then don't you worry. You haven't even reached it to any nearly enough to that any limit of like arriving at such the possible variation limit, so to speak. Here, okay, so having said that, you have 3 billion nucleotide on your genome, but like I said, not all of those 3 billion nucleotides act as DNA. So in other words, what you can say that only certain region, not all, but only some of them, only actually very small area, only very certain region of DNA act as genes. Okay. How do you know? Like this 3 billion base pair of human genome, then after the human genome project completed, later actually I will make some kind of addendum at least, so that we can briefly go over what's the nature of human genome project and how it is being done sometime somewhere in this chapter. That's what I'm going to do. But it turns out that we do have even less than 23,000 genes present on this long DNA nucleotides. So by simple calculation, that means if you simply divide this 3 billion by 23, then the number of genes, that means by calculation on theory, like the length of average gene would be 150,000 base pair long. Okay, if every area of your DNA is taken up and used as genes. Yeah, that's like very simplistic conversion calculation. However, in reality, actually average length of gene, as it's been long known that on average the length of genes in mammals, not only in human but in all mammals, is only about 30,000 base pair. So it's 150,000 versus 30,000. Even this 30,000 base pair is this really conservative estimation. Actually, usually it's not even 30,000, but the region area where out of that actually the information of the protein in terms of which amino acids are where, like the final product of the gene, the protein, then it is not 30,000 but something like usually only less than 5,000 base pair. So there's a huge discrepancies between the calculated things and the actual reality. Clearly indicate that not everywhere on your genome is actually being used as the area like defining the gene. So with that, actually what you can see in terms of the description of the structure. If you in the chromosome, if you untangle the chromosomes so that you line up all your DNA in a single, like a DNA strand like that, then certain area can function as a gene, followed by the area which does not do anything in terms of like functioning as a gene. Those area we usually call as an intergenic region, and then followed by a gene. And in reality, the area where the DNAs are used as genes are much smaller in terms of their area than this intergenic region. In other words, majority of the area on our DNAs are doing nothing. So in the earlier days of this discovery, scientists used to call this intergenic region where no gene functions are provided by them. So the scientists call this area as junk DNA. But then later, of course, scientists knew better that, oh, this is not simply junk, but there are all these intergenic regions, even though they do not act as actual genes, but they do have very important functions. Although it is not really unified function, nor something that we understand their function clearly enough to explain what they are, but at least what they know is, okay, these are not simply they are junk, just like we can remove them and then live happily ever after. We still need those so-called junk area of the DNA in our genome too, is probably at least a temporary conclusion at this point. Okay, so we can at this point define the gene. So what is gene? The definition of gene actually has a relatively long or a longer history. So from classical genetics point of view, the genes, the classical genetics, by the way, this classical genetics called the transmission genetics, which was established and found by Mendel, Gregor Mendel. This classical genetic point of view, the genes are defined as some kind of factors that determine the characters. What's characters and factors? Viewing the gene as some kind of factors, it is an individual and separable kind of unit. So earlier days is something like 1800 or something, where that renowned Gregor Mendel, who was the founder of this classical genetics, he viewed this gene. There was no actually clear definition or understanding about what genes are, but then for the first time, this Mendel thought correctly about what genes are, and he thought genes as some individual unit of factors that can determine the characters. What's characters? Did I actually already go over the difference between character and trait earlier? I'm not really sure. I don't really remember, but yeah, I will do it again over here. So character is some kind of the characteristics which we believe that which is determined by each different gene. And under such one category of character, you can have several alternative versions of sub-characteristics we call as trait, right? So under a certain character, which is defined by a gene, several different traits usually exist. For example, flower color. Speaking of flower color, flower color is one such character, right? But then flower color can be different, many different flower color, like red, white, yellow, and blue. Even in a single species of flower, certain plants, same species of plant, have a red flower and another one is a white flower, for example, at least. So those are the different traits on the single character. You can imagine so many other such examples of a character and under which many different variation of the character in different traits. So later we could hopefully understand better about this actually nature in terms of these molecular nature of this character and the trait. However, for now, at least Mendel, at the time of those Mendel, he was the first one who correctly viewed these differences. So the gene is the one defining character, and under certain gene you can have several different alternative version of the gene. And we know actually if the character was determined by the gene activity, then you know that we are, at least human and most other organisms, are a diploid individual. So in terms of a trait in a given person A have two chromosomes. These two, like two copies of a chromosome. These two copies of chromosomes is called homologous. Chromosome, right? In Korean, it's called this. Why do you have these homologous chromosomes for each unique chromosome? I have already mentioned this, covered in earlier chapter. One chromosome number 1 came from father, and another, the partner chromosome number 1 prime, came from mother, for example, and they are basically the same chromosome. So if the chromosome is the kind of vehicle or container for genes, then at that same position, the same type of gene or character can be found, right? However, the thing is, at the same position, but actual nature in terms of a trait could be different, right? So this is a trait a little bit different from this is the nature. So you do have character defined by gene, but depending on the actual DNA sequence variation, because if this chromosome came from your father and if this chromosome came from your mother, and your mother and father has been maintaining respective their lives independently and their ancestors too, so there is no reason why their DNA sequence should be identical in any given region due to the mutation, right? So then there's no reason why you can simply expect that their DNA sequence will be 100% identical. So as a consequence, you may have a little bit of the difference represented in different traits. So that's the nature, and miraculously, without having any slightest idea about what exactly the actual nature of DNA and chromosome, Mendel, even in 1866, in such an early date, knew kind of, at least in conceptually speaking, with such an accuracy, he understood correctly about this nature of this. So the discrete means he established this concept of the genetics. So it's kind of a separable and unique discrete factors as a gene. So his experiment actually involved with in pea, garden pea, scientific name is Pisum sativum or whatever. One such character, flower color and flower position and seed color, shape, pod shape. So all these are different categories of characters, each of which is determined by different genes. But then, as a diploid organism, P also generated two different alternative forms, the purple flower versus a white flower, both of them examples of a trait, right? And also whether and which position this flower is attached. The flower could be attached on the terminal or sideways on the axial. Both of them, both conditions are an example of respective traits under the heading, under the category of this flower position, and so on. And the nature of the why you have such a different trait is because it's a different organism and diploid. Each different organism under the same species, they are both diploid, which means they have two chromosomes, each of which contain particular state of the gene. So like this one individual have two such chromosomes having one specific status of a gene, whereas another individual have this particular status of a trait under the same category. And during the gamete production, you have, for example, here you have, there is no other choice but you will produce this gamete, and you will also in this individual will produce this gamete, and these two gametes, haploid gamete, will be combined after during this fertilization. Now you have this combination, the mixture of the two different traits exists over here. What happened is, and then the traits outside the apparent trait of this individual will be that of this particular one having this particular state of this gamete. Why? And since this is not a general biology course, so I will not just go into too much exhaustive details about this like hybrid production and following that mating in the next generation, which Mendel did, and then figured out and predict. He was able to predict in the next generation, the first generation, which is called the F1 generation, what type of traits will appear. All of them, actually, in all the offspring, only one kind of trait appeared between these two different traits of parents. But then further continuation of such mating, this was actually self-fertilization. Without inviting any other individual having different traits of this character, Mendel simply continued the reproduction within themselves of these F1 generations. That is a self-fertilization technically. And then he obtained this time in the next generation, which is the F2 generation, he was able to obtain two different kinds. Now, one kind, which is the type of this particular parent, and another is type the trait of the other parent, original parent. And actually the importance is he was able to predict the ratio. How many of those offspring in the F2 generation will have this particular trait, whereas the other type of traits, how many of them will be produced type of. Why? Because mathematically speaking, if his idea was based upon this separable. In these days, we know this separable thing is an gamete, okay, and which is like haploid chromosome in each different gametes, like a sperm or an X, whatever. And then those haploid gamete will be recombined during the fertilization, and then now this chromosome will exist in different combination, and then if you keep doing this, then a new combination will be generated in the next generation. So this is basic concept of genetics Mendel figured out, and that was really remarkable job, which was the start of this genetics. Although in modern genetics, which was taken over by, this was a classical genetics, but in these days of genetics was largely taken over by so-called molecular genetics, where actually people understand the actual nature of this whole genes interaction based upon its DNA and its actual DNA sequences and all variations like that. So even these molecular genetics are prevalent in these days, still what we need to understand is some of the terminologies, okay, came out of the classical genetics originally from Mendel's work, which is this. Although here I have provided some terminology definition based upon such classical genetics, okay, I can actually have another way of explanation of this using this diagram. So, for example, like genes, although we are going to define the gene shortly after this, genes in terms of this molecular biology or molecular genetics, but purely based upon the classical Mendelian genetics viewpoint, we all know that genes are on the chromosomes. Yes, genes are located on chromosome, although Mendel himself in the beginning of this Mendelian genetics, so there is no any kind of, nobody knew what's the relationship between genes and chromosome. There's no like idea. People didn't know what chromosomes are. But anyway, what we know in these days are genes are on chromosomes, right? And so since genes are on chromosome, and then we all know also that not every location of the chromosomes are occupied by genes. So then some area, for example, on the chromosome here, this is a chromosome, but this is one such location of one gene, and another location, and another gene's location, like that. And so each of these location of the genes on the chromosome is called locus. And plural form of this locus is a loci. So 3 locuses. So we now call these 3 locuses a loci, 3 loci. And each locus is the location, position of these genes on particular chromosome. So that's the explanation and the definition of this particular term, locus, which has been frequently used in Mendelian classical genetics, locus too. Now another terminology originated from Mendelian genetics is these alleles. Okay. So for each genes on a particular location, so this is one locus. But then this locus, the genes are in different color coding intentionally, right? So this is one character, but 2 different traits. One trait is orange, and the other one trait is yellow. Okay, maybe eye color. One eye color is orange, and the other eye color is yellow, for example. It is possible, right? Because you have 2 chromosomes, right? 2 homologous chromosome set. Each locus you contained one same character, but the actual nature could be slightly different due to the DNA sequence variation. So anyway, so then this alternative version of any trait is called allele. Okay. Here we have 2 different alleles. Sometimes these 2 alleles could be different. Different, and some other times it could be identical. If the alleles on the specific locus is the same, then we describe such a status as a homozygous, the same, homozygous. But the problem occurs when they exist, these 2 alleles exist in 2 different. If the alleles on the same chromosomal locus, which is under the same character, are different, then we describe that as an heterozygous. Now you have a problem. If these 2 traits are in heterozygous status, then which one? Let's say this is an eye color. One eye color trait will signify the orange eye color, and the other alternative allele will signify yellow. Which one wins is the problem. since you have two eyes, so left eye will be in orange and the other eye will be yellow, but that is not the case. Although, yeah, I know that some mammals and some human can have this two different eye colors, but that's something different from this. And besides, human eye color is not determined by a single gene. Actually, it is a result of several interaction of many different several genes, and then define one single such character. This case, but anyway, in case of this heterozygous, only not kind of mixture form, but either one of them will express itself to outside. So to outside, in terms of a particular trait, only one of these two will show. So the trait show outside, we call it as a dominant trait. The one, even though it exists, but it's hidden in a heterozygous status, we call it as a recessive. So that's the simple the reason or the relationship between dominant and recessiveness. The one shows up is defined as dominant, and the one hidden is defined as a recessive. Okay. Why something is dominant and the other one is a recessive? Okay, what is dominant? Why is two different animals. If you were asked what's dominant and what's recessive in terms of its trait, then you simply explain. Your answer should be, well, in heterozygous status, if a single allele, one allele, shows up to the outside externally, then that is a dominant allele. And if the other one is hidden, is not expressed outside, then it is a recessive. Then that is the answer to the what is dominant and what is recessive. Okay. By the way, all these are the dominant and recessive, whatever, all these alleles are result of mutation. The reason why under a certain character, which is defined by a gene, one single gene, the reason why you have many alternative version of these alleles are all result of mutations, right? So, for example, in this individual, how many different alleles are possible for this individual A? How many different alleles? The maximum number of different alleles in this individual is only 2, because this individual is a diploid, right? However, if you ask the same question like differently, how many different alleles are possible per each character? Which is asking how many different way of a mutation, version mutation is possible for a single gene. Answer is countless, limitless, right? Because you can have so many different ways of making this DNA sequence change, and potentially each DNA sequence change can become a new trait or new allele. So that's the kind of difference. So getting back to this, the dominant and recessiveness. So we have already defined what is dominant and what is recessive, and both of them are dominant alleles and recessive alleles are all the consequences of mutation. Now let me ask you that why a certain allele is dominant over the other, which is a recessive. Then the answer should be something different. Why? When you were asked why, which means dominant. If you think about it, dominant. Let's take a look at the recessive. Try to define recessive allele first. Remember, all alleles are result of DNA mutation. So it's basically same thing to ask why certain mutation is recessive and whereas another mutation is dominant. In the case of recessive mutation, in other words, recessive means even if you have this particular conversion, you are irrelevant in the heterozygous status because the other allele will simply cover your consequence. Why is that so? It's all about the function. Like if this is a loss of function, if this mutation result in the loss of a function of original thing, then it will be a recessive mutation because as long as the other already existing functioning version of this gene exists in the form of a heterozygous, then that functioning one will cover up your loss. Don't you worry, I will take care of this function. I still can function. So even if you lost your function for now because of this unlucky mutation, so to speak, doesn't matter. Doesn't affect the individual who carries this loss of function mutation. In that case, that loss of function mutation will become recessive, right? However, in the case of dominant allele, this dominancy will come up then if the mutation will result in any gain of function, gain of new function, then the existing one cannot do anything about this newly came up function. Well, if this new existing function can somehow have an interfering role, then that is a totally different story. Then now we have a whole new mess up thing. But in the case of a sure gain of new function, then this will show up because there is no way you cover up a new function because you haven't seen this thing before. Okay? So in the case, it will become a dominant. And once again, also, if this new mutation is somehow kind of or interfering existing function, then also it will become a dominant one because there's no way you can hide this thing, cover up in the case of this loss of function. So that's the reason, and that's the reason why certain mutations are recessive and certain mutations are dominant. It is not, maybe I don't really think in these days you have a kind of a confusion about dominance versus recessiveness is actually the superior and an inferior kind of relationships. Don't get confused with such thing. Just because you have a dominant allele, that doesn't mean that dominant allele is something superior in its function, superior over a recessive one. It's all about this molecular mechanism of this mutation nature. Okay? And speaking of such dominance and recessiveness, once you understand this actual nature of dominance and recessiveness, then you can also kind of understand that not always you have a clear-cut dominance and recessiveness actually established. Sometimes it is a kind of a shade of gray, so there isn't actually clear kind of a distinction. I can show up and I can suppress this expression of the other guy. Sometimes you can both of them can be expressed. Sometimes like kind of a mixture looking like a kind of a in between, halfway between of this defect can be expressed outside. In those cases, it's called the incomplete dominance. The former case that I just mentioned, and then it is called the co-dominance. Later, when we when I introduce the example of the AB blood type, we can also have a better understanding about nature of this codominance. But anyway, so that's the kind of summary of terminology and concept. Those terminology in the modern genetics, which is molecular genetics viewpoint, the concept and the reason why those terminologies occur is what I have tried. So with that, time has already been something like way over 1 hour and 30 minutes, so I will have to stop at the first session of the lecture for now, and then the following session I will move into now the modern molecular genetics of this whole aspect.